experimental design, and it's going to touch upon many of the points already raised, both in the presentations and in the discussions, but nothing wrong with that. First, I wanted to make an important announcement, which is that September 19th, which is Thursday, I think, is International Talk Like a Pirate Day. And what do pirates say? Exactly. It's a significant date for this course. We'll talk a little bit about designing experiments and elements of experimental design, the kind of decisions that you have to make when you yourselves design an experiment, or what you can incorporate if you're doing an observational study instead. One of them, of course, is plan your sample size. So you're all planning studies. How do you know how many individuals to take? And one of the reasons why this issue is sort of at the forefront, again, of discussions about data, is that a lot of experiments, a lot of studies are underpowered. And that seems to be one of the main reasons why replicability is low. In the UK, though not yet here, in the UK when you apply for a grant, you have to justify the sample size as part of the grant application. So you have to basically include a power analysis or some sort of analysis that helps to justify the number of animals you want to kill or otherwise abuse in your experiments. And anyway, science is expensive. You don't want to overdo it. So there are some issues with studies that have low sample size or low power. And of course, the one we're always worried about is that if there's a real failure, we'll fail to detect it. And that's generally the one we're most worried about when we design an experiment. But there are other issues, as I just said. Low power studies tend to have low power, and that means the probability of getting the same result again if you repeated the experiment is low. And this was regarded as perhaps the main reason behind the low reproducibility of the study that I talked about in the very opening lecture, where the publication in Science, where a group of people repeated 100 psychology experiments. Maybe less appreciated fact is that low power studies that do reject the null hypothesis, maybe false positives. And a somewhat counterintuitive one, but if a study has low power and a statistically significant result is obtained, there's a fairly high chance that it's actually in the wrong direction. Low power basically means that it's almost a fluke if you get a significant result. And that fluke could happen in any direction. Finally, if your study has small sample size and the estimates or effect size are going to be highly uncertain. And for many purposes, we're not just trying to get a significant result, but we actually want to know something about the magnitude of the effect. And that's not very easy to estimate if sample size is low. So these are issues that we confront when we think about designing our own experiment. One that a problem that I also brought up in the very first lecture with reference to this 100 repeat studies is that if a statistically significant result is obtained in a study that had low power, the effect size estimated is going to be exaggerated, almost with a high probability. So in some areas this is called the winner's curse. And generally, the magnitude of the overestimate that you get depends on the power of the study. So if you have an exceptionally low power study and you get a significant result, you basically need a fluke result, and that fluke, of course, is going to be an overestimate of the effect size. And as I mentioned, also it might actually be in the wrong direction. So in this publication, they plotted statistical power and calculated the magnitude of the bias on average. The magnitude of the overestimate of the effect size. And so you can see that, at low power can be associated with substantial bias. And in this particular review, they attempted to estimate the power of most studies that have been published. And they concluded it was somewhere between 10 and 30%, suggesting that most results in neurobiology are between 20 and 50% overestimated. So when you sit down and figure out what kind of a sample size that you should have or use, there's sort of three considerations that you might think about. One is, you might be trying to estimate a magnitude, a number that's important. Is it large or small? Is it important or is it trivial? And in that case, you would plan for precision. You would design an experiment not only to get that magnitude of interest, but you would want to put bounds on that estimate that are not too wide. A 95% confidence interval is this big. Whether or not you have a statistically significant result, a confidence interval this big means you have no idea what's going on. Whereas a confidence interval that's relatively narrow at least allows you to say, well, with reasonable confidence I can say that the true effect size is somewhere between here and here. That's what we mean by planning for precision. And we're going to do an exercise like this in the workshop on Thursday. Planning for power you might be more familiar with, and basically it's to say, you have to say, all right, let's say the effect size is this big because so-and-so found it to be approximately that size. And that's what I would expect to see in my system. What sample size would I need to reject the null hypothesis with 80% probability for an effect size of that magnitude? And I'll show you in the lab on Thursday as well some tools in R that will calculate those sample sizes for specified power and effect size. And finally, because we work on animals and plants, some of them are going to die. And so take that into consideration as well. You may end up with a larger experiment at first, but then becomes reduced in size through no one's fault, really. And then there's hard decisions about sample size. Should you have more plots or more plants per plot? Should you have more families and fewer individuals per family, or should you have few families with many offspring in each of those families? Which of those would produce the more powerful study? Which of those designs would produce a confidence interval that's as narrow as possible for a given sample size or allocation of individuals? And so again, there are tools in R for answering these questions. And actually, one of the best things about R in attempting to decide these things is that you don't actually need to find a formula. You can just say, here's the effect size that I'm imagining. Here's how much variability I expect based on my reading of the literature. You can simulate your scenario, even if it doesn't fit into one of the standard, two-sample designs, to allow you to answer questions like this. So we've done this in my lab to answer exactly that question. Should we have more families with fewer individuals per family, or should we have fewer families with more offspring? The answer is usually more families with fewer offspring. It depends on the scenario. And R is just, once you become handy, and I'm sure you will, with R, you'll realize that it's possible just to simulate data and answer those questions. And one of the exercises we'll do on Thursday actually involves a simulation, a very simple one, but to get you sort of thinking about what you can do with R. Okay, there are challenges. Some of the magnitudes are unknown to you, like how much variability there is. And so you have to make an educated guess, or you have to go to the literature. Now you know that literature is biased. Probably the variability is greater than in those few studies that managed to get a significant effect size. So keep that all into account, that the literature itself might be biased. But maybe a previous experiment has been done in your lab already, or maybe you should carry out a pilot study, at least to try to measure some of those things. We're going to do a little bit on post hoc power calculations, something that was once done with frequency, but that was done routinely, which is to say that you do your analysis, or you do your study, you do your analysis afterwards, you fail to reject the null hypothesis, and then you say, yeah, well, I didn't have much power anyway. And you try and calculate what that post hoc power was. So this was already touched upon, but I want to go over it just to make sure it's absolutely clear. What's an experimental study? The essence of experimental study really is that there's at least two treatments, and that the researcher assigns treatments to the units or subjects. So that is the definition of an experiment. And Hurlburt refers to mensurative experiment. That's not an experiment at all. I would just sort of abolish that term. That's an observational study, just a very complicated, no matter how complicated it is, if all you're doing is measuring something, you're not doing an experiment. If you're sequencing genomes, even as many as 3 of them, you're not doing an experiment unless you're actually assigning genomes to individuals. So there are classic examples of experiments: clinical trials, reciprocal transplants, factorial experiments on competition information, and so on. And observational studies that would then include common garden experiments, QTL experiments, and so on. But the majority of those do not have 2 treatments.
What about QTL experiments?
Oh, sorry, this is a genetics method for mapping traits to genes. So why do experiments? This was mentioned briefly in the presentation. And the reason is that an observational study can't distinguish between 2 possible explanations for an outcome, an association between an explanatory variable, X, and a response variable, Y. So Ray Hu did a bunch of analyses of data on people who climbed Mount Everest. And people die climbing Mount Everest, so there's a rich data source there if you're interested in fitness. And usually, apparently, people die on the way down rather than on the way up. And it's been shown, Ray Hu showed, that for those individuals that brought supplemental oxygen, their probability of surviving a climb of Mount Everest was higher than those individuals that did not. And that could be for either of 2 reasons. One, supplemental oxygen is actually good for you, and there are physiological reasons to believe that. But the other possibility is that, that's just indicative of greater overall technical preparedness for the climb, and all of those other variables that are associated with supplemental oxygen also might be solely responsible, or at least make a contribution. And so bias our estimates of the true effect of supplemental oxygen. And all those other things are called confounding variables. So what an experiment allows you to do is it allows you to randomly assign treatments to the subject. And that's the only way to tease apart the effects of the explanatory variable of interest and all the confounding variables. So if assignment is random, there will be no confounding variables associated with the treatment of interest except by chance. So in this context, the way to do an experiment is that you, the experimenter, decides which teams are allowed supplemental oxygen and which teams are not. As you might expect, the volunteers might resist this treatment, which of course is why experiments on humans are so challenging and even. But the point is that by randomizing, you break the associations with confounding variables. Clinical trial. I want to mention clinical trial for a couple of reasons, and one is that people have thought a lot about experimental design in the context of clinical trials. Why? Cost of making a mistake is really high. People think very carefully about sample size. You don't want a larger experiment than you need, and so on. So clinical trial is an experiment on human subjects. Other experiments that we normally carry out would be lab experiments or field experiments. And when we were writing the book, we were looking for, what would be an example of a clinical trial that would at least demonstrate some of the features of clinical trials and allow us to think about them and the effects that they might have. And so we chose this particular human trial, and it was about a gel that in the lab it showed promise as being effective against transmission of HIV. And at the time of this experiment, HIV transmission rates in Africa were really high, particularly among sex workers. And there had been laboratory experiments to suggest that this gel was actually effective. And so there was a sufficient amount of evidence had accumulated that caused people to say, okay, let's just try this in reality. And it's amazing to me that experiments like this go on, that it's a true experiment. They managed to gather a sample of 765 HIV-free sex workers in 6 clinics in Asia and Africa. And they randomly assigned 2 treatments. Either they got the gel or they got a placebo. which is defined as an inactive compound that subjects cannot distinguish from the treatment of interest. A feature of this experiment, besides its size and that it was carried out on humans and on a population that a large fraction of which would eventually acquire AIDS, was that nobody involved in the experiment actually knew which subjects received which treatment. So a system of numbered codes kept track, and only the statistician knew what those codes meant. Here are the results of the experiment. So the gel and then the placebo. Here are the clinics, the number of individuals in each of the clinics, the number of individuals that became infected out of that larger sample. So during the course of this experiment, a substantial number of individuals acquired HIV. They're all sex workers in these countries and visiting these clinics. And the overall finding was that the risk was the same in the two treatments. This was totally ineffective treatment as far as the experiment was concerned. If anything, the HIV rates were higher in the treatment category than in the control category. So what are some of the features I want to point out from this experiment? One is, okay, there was a control, a placebo. The second is, crucially, the treatments were assigned randomly to women. And second, or additionally, nobody knew which women were assigned which treatment. There was absolutely no way that any kind of unconscious bias in the people who were evaluating and testing these women as they came repeatedly to these clinics knew who had received what treatment. And that is called blinding. And of course it was replicated, it was relatively balanced, and it also involved blocking. And blocking was something that was mentioned in the presentation. It basically amounts to repeating the same experiment again and again in different locations or in different sort of groups. So why tell you about clinical trials? We don't work on humans. Well, because there's a compelling argument that we should be carrying out our experiments pretty much the same way that clinical trials are conducted. That we should attempt to include all of the components of clinical trials, even though we might be working on plants or animals or bacteria. So in this survey, which was published in PLOS Biology a few years ago, they carried out a survey of animal experiments and just reported on a variety of features of the published literature. Only 60% of the 271 articles stated the hypothesis or objective of the study. What? Don't do this. Okay? They're not, especially if I'm on your committee. Number of animals used is often not reported, or characteristics of the animals. Few of the papers surveyed reported using randomization, random allocation of treatments to subjects. Few of them reported blinding. 13% reported blinding. No, 14% reported blinding. The statistical methods were not described adequately in 30% of the publication. So that was a topic that came up in our discussion. Should you trust a paper that doesn't give you enough information on the statistical analysis? So it would be useful, because there are such strict and adhered to guidelines for clinical trials, experiments on humans, maybe we should bring some of these features into our own experiments. So that was the argument of this paper. So we all know why we need a simultaneous control. Because, well, we need a control. You know, I mean, particularly human subjects, this is important because human expectations of treatment often affect individual health, particularly for measures that are self-reported. And you know, that's known as the placebo effect. It's been shown over and over again that placebos actually work. They're effective. Not only that, they work even if you tell the person that they're getting a placebo. There's something weird about our brains that, as a result of receiving attention, actually improves our health even when we're told we're getting sugar pills. Go figure. But something that we don't often think about, or maybe not enough, is that the control should be abused in the same way as the experimentals. And for field experiments, that you know, in physiology, that might be a sham operation where you carry out pretty much the same operation, but you know, missing one last detail, i.e. the treatment. But in field experiments, you know, that might just involve trampling the control plots as much as the experimental plots. Randomization. Well, randomization is necessary because that's the only way to break the association with all potential confounding variables. And it has to be done with a random number generator. Humans are terrible at coming up with a list of random numbers haphazardly. Almost any series of numbers that you can come up with, I could show you significantly different from a random series.
Well, if in case you don't have a computer, would you
Just flip a coin.
What do you mean you don't have a computer? I see a lot of computers here.
No,
You should not go out into the field until you've chosen your random numbers, I guess. I like that part of your design. Actually, you know, you can also record the random number seed that are used when generating numbers. That allows you to generate the same random numbers all over again. So it's really important to know that randomization just breaks the association with confounding variables. It doesn't eliminate variation caused by those confounding variables. It just evens them between the treatment of interest and the controls. Factoid, a completely randomized design is one in which treatments are just assigned to all units by randomization. And I'll contrast that with other possible designs shortly. So blinding is the process of concealing information from participants. And not just from the subjects, but even the researchers who are measuring those participants. Because unconscious bias can slip in no matter what. How are you feeling today? How are you feeling today? So just the possibility that researchers might clinicians might treat subjects differently who are receiving the treatment versus the placebo means everybody must be kept in the dark. So an experiment is double blind if neither the subjects nor the clinicians know who's receiving what. Single blind just means that the subjects, the units, are kept in the dark, but the clinician knows. So there are meta-analyses that do things like, you know, show that on average experiments without double blinding are more likely to exaggerate the magnitude of experimental effects and so on. But these meta-analyses, although they're instructive, there are usually confounding variables. So studies that don't have blinding, you know, also are missing other things, and so it can be difficult to figure exactly where things went wrong, because they often go wrong in multiple ways. So here was a survey of 292 treatment experiments carried out on animals or cell lines. And they found that the odds of detecting a positive effect of treatment were threefold higher in studies without blinding than in studies with blinding. But again, other things there are correlated wrongs that go with this particular wrong as well. You can incorporate blinding. Of course, it doesn't really matter what the plant or animal knows that you happen to be working on. So, but you can do what you yourself know. If you're, you know, measuring things, then you can, you know, not group them into two piles. Measure one pile, the treatment, and then measure the other pile, the controls. But actually, use a system of random numbers to identify them, measure them in random order, and then figure out later which treatment the units belongs to. That's not reported very often in our field, but I think it's a very useful step, and will keep you out of trouble. Sampling error, well, that's why we replicate in the first place. We want to minimize background noise. But there are a variety of ways to minimize background noise that might interfere. And one is to, you know, make the experimental conditions as constant as possible, humidity, temperature, and so on. And I mean, there is this worry that maybe constant conditions aren't always desired. We would like to know, as was mentioned in one of the discussion comments, that we would like our results to be robust. We don't want them to hold under very precisely controlled experimental conditions that they become irrelevant in the field. Certainly in human studies, controls, I mean, one of the justifications for carrying out most clinical trials on men rather than women has been the need to control everything because men and women respond differently. And NIH now mandates that experimental designs must, henceforth be carried out on both men and women. And I read a recent report about this, but there are ways out of this. For very good reason, if you have very good reasons, you can still do it primarily on one sex, and generally the chosen sex is male. But nevertheless, it is advantageous to minimize potential background noise. Another way to do this is to use extreme treatments. You want to really notice something, make the effect big, at least the first time you do the experiment. Replication is one of the ways we try to minimize the effect of background noise. So more units is always better. Large sample size is always desirable. And we've already had a discussion about what replication really means, and that was the main reason I made you read this long paper by Hurlburt, which is still classic and, as was said in the presentation, still relevant. Here are 3 experimental designs. We would probably never carry out this kind of experiment, but actually we would sometimes carry out this experiment. I've seen it happen. One of my students did this, and there were constraints that prevented him from doing the experiment in any other way, but the result was that at the end of the day, all he could say was that this chamber was different from this chamber, but not that this treatment had an effect different from that treatment. So that's the if there is a constraint, that's the consequence of that constraint.
But, sorry, Dolph, couldn't you, even though you have that constraint, couldn't you say there was a difference in treatment? Maybe not what your student did, but is it
Well,
possible?
the trouble is, statistically what you're showing is that there's a difference between chamber 1 and chamber 2.
Right.
Now, you hope it's because you did treatment A in this chamber and treatment B in that chamber, but how do you know?
But is that still good science then? Like what?
Well, it's unreplicated science. And so you have carried out an experiment with 2 treatments, but you haven't broken the association with confounding variables. Chamber is a confounding variable. So essentially, you've not carried out the randomization part of experimental design.
So if you ran the experiment end to end in just one chamber, would that get around that, or would that just
It would.
use a
It
different
would
kind of-
potentially get that around that. That's correct. So here's making some of these points again. But something I want to point out is that an experimental unit doesn't have to be a single individual. It can be a group of individuals, such as a field plot that has multiple individuals, a family that has multiple offspring, a petri dish with multiple colonies, and so on. The key thing regarding the topic of the paper, pseudoreplication, is how you analyze this. And how you analyze this is something that we'll learn more about, and we'll be using the method in R. Someone mentioned using a nested analysis of variance, and that's an old word for what we now call mixed models, and that's exactly correct. That once you take the structure of the design into account in your analysis, then you're fine. You're not pseudo-replicating anymore. Right. So this makes that point. Multiple subjects or individual organisms belonging to the same unit aren't independent. So you can't treat them as though they are. You need to take the structure of the design into account. And one of the themes, I guess, of this lecture would be that analysis follows design. So keep in mind the design, and your analysis should mirror it. And so this is the pseudo-replication mistake: is pretending that the individual organisms are your replicates when they are grouped. Balance, I don't have to spend a lot of time on. Then we'll get wrong here. That's a closed parenthesis. That's an open parenthesis. Balance is actually generally good, and it's a feature of this clinical trial. A couple of reasons. You actually get more power and more precision from a balanced experiment than not. You know, this quantity, which figures in the calculation of the standard error of the difference between 2 treatments, is actually smallest when n1 is equal to n2. So That's a reason. But balance has other benefits. Generally, your ANOVA is more robust to departures from normality if you have balance. So it's something to strive for, but it's not the most important thing. Blocking. So that was also mentioned in this experiment, the clinical trial that I described. Blocking was basically repeating the experiment in multiple clinics. And so, as I said, repeating the same completely randomized experiment multiple times, once for each block. And here's a way of doing a replicated experiment when you have only 2 chambers. So try and randomize the order or placement of these things within chambers and apply the same experiment repeatedly, once in chamber 2 and once in chamber 1. And now we have replication. Now, analysis follows design. We need to take the fact that there are 2 chambers into account when we conduct our analysis. And that's what mixed models are for, and we'll be looking into that. Another way of minimizing the influence of sampling error is to use a paired design. And actually a paired design is a kind of blocking. You've all done paired t-tests in your undergraduate days. And you think of a 2 treatment design where you're applying different treatments to field plots, and you have a choice. You can apply the treatments randomly to different plots, or you can divide the plots and apply both treatments to every plot, but one on each side, randomly chosen. And the idea is that if there's spatial variation so these represent blocks, and if there's spatial variation in potentially in the response variable that you're measuring, say plant growth, then because of spatial variation in nutrients or light or whatever, the paired design will cancel a lot of that variation, and so the treatment effects can stand out. So there are very good reasons to use blocking. You just have to make sure that there's a really good divide between the 2 halves of your plot, so that the nutrients or water don't leak from one side to the other. So the paired design is just a special case of a blocking design called the randomized complete block design. But if you had 3 treatments per block or 4 treatments per block, that would be sort of a next level up from a paired design, and that's called randomized complete block. So in our example clinical trial, clinic is a block. I just want to mention experiments with more than one factor and why they might be useful. You've all heard of factorial designs. It just means you have more than one treatment. And factorial design is an experiment in which all combinations are included in the experiment. And you usually do this because you're interested in interactions between those potential causal variables. And even if there aren't interactions, a factorial design can be an efficient design for gaining information about the effects of 2 different kinds of treatments, all in one experiment. I can't remember why I stuck this slide in here, but it's a warning about replication in the specific case where time is one of the factors. So I've read a lot of theses and read a lot of papers in which a main treatment of interest is applied. In this case, it was sort of presence or absence of rodents on desert plots. And the response variable was the number of ant colonies on these different plots. And there were actually 2 plots of each treatment. So this is n equals 4 plots. But the number of colonies was measured multiple times, allowing the inclusion of another factor called time in the analysis. And it's a common way of analyzing data, and it's a common way of making a mistake when analyzing data. So in a mixed model, which we will learn, the structure of the design says that we have 2 plots where treatments was applied and 2 control plots, and we're measuring those plots repeatedly. So in general, there's a way in which we can nest or use a mixed model that takes repeated measures into account. However, time is additionally problematic for another reason, and that is that adjacent points in time will tend to be more similar. The residuals will tend to be more similar when the same plots are compared adjacent in time, then more distant in time, and that violates a little-known assumption of analysis of variance as well. So I'm not going to go into great detail other than to say, warning, warning, time. Time can cause issues, even when included explicitly in your design. How do you decide the best design for your purpose? Well, there are tools out there. There's actually an R package, Agricole, which will help model a lot of the designs that people typically use in agricultural studies, such as completely randomized, randomized complete block, balanced incomplete block, split plot, and so on. So these are all conventional designs, many of which were designed for agricultural studies, and many of them are translatable into other kinds of studies, not necessarily agricultural. If you try it, please tell me. I'd be interested in knowing what your experience was, or whether you gave up and not just simulated. So one of the last points I want to make about design in general is that when you analyze the data, the analysis should reflect the structure of the study design, and we'll learn ways to improve. We'll learn some tools in R to help with this. So pseudo-replication really is a problem of analysis, not of design. It happens when the structure of the analysis doesn't match that of the experimental design. So the most common mistake is that subjects are grouped, and the analysis then leaves out the grouping variable itself. So you carry out an analysis where your experiment occurs in blocks, for example, but then you leave block out of the analysis. So the best thing to do is to employ that structure when you analyze the data. And I would actually recommend analyzing your data before you have it. So I would recommend that when you design an experiment, that you make up data and actually try out the analysis you're thinking of using to get some experience with how it works. And that would also allow you to do some simulations to figure out, to figure out answers to decisions like, should I have more offspring per family and fewer families, or vice versa. Anyway, R is good for making decisions. So I want to touch upon what if you can't do experiments. And a lot of us do comparative studies, observational studies, and certainly no one's going to do an experiment until you've done an observational study to suggest that maybe some factor of interest is important. And so what do you do in this case? And the sort of best practice of procedure would be, well, incorporate as many of the features of a clinical trial as you possibly can. The one thing you can't do is randomly assign treatments to units. But you can do most other things. You can do blinding. You don't need to know which plot the observations, the plants you're measuring actually come from. And there are other ways that I'm not going to describe, but what's commonly used in observational studies on human subjects is matching, where there will be two treatments and individuals are sort of paired by age and economic background and so on when the study is done. Essentially, blocking is done by bringing individuals into the study in matched pairs. Okay. I talked a little fast. It's only 1:45, but that's okay. Maybe there's some cake left. Here's the discussion paper for next week. So it's going to involve some themes that we'll hear about in the future, and also about power calculations in general. And I have one volunteer already to present. And so if you would like to present, come on up at the end of this lecture. And I'm also looking for 2 moderators for next week. And as I've said before, if not enough people come up, I'll be using the sample command in R to choose one of you to present or moderate this week. And we'll be using the sample command in the Thursday workshop. I'd like to get a sense of how people are doing so far in the R workshop.